This video is created to accompany access to the Genomics England Rare Disease Project and the Nextcode Health Clinical Sequence Analyzer. Its intent is to demonstrate how to reach the conclusions presented by Nextcode to the 2014 Genomics England Rare Disease Bake-Off. At the Nextcode Health website, please log on to the customer login using your email and password. There are 5 disease cases provided by Genomics England for interpretation. Under the Studies tab, you will find a list of the studies provided. They are labeled from 12 to 16 and are selected individually by clicking the link. Study 12 is a trio consisting of unaffected parents and an affected male offspring. The affected child has Ohtahara syndrome or early infantile epileptic encephalopathy. The phenotype is further described under description. Entering seizures in the dialog box labeled Phenotype Signs and Symptoms populates the candidate gene list with 388 genes that are associated with that HPO code. To view the candidate gene list, click on the Candidate Gene Overview button. In each of the cases presented here, we will proceed in CSA using default analysis parameters other than including paralogs of candidate genes. Analysis parameters are manipulated in the Report Settings tab. Default settings are maximum 1% allele frequency and 0.1% genotype frequency, and variant effect predictor consequences of high and moderate. See training videos for advanced parameter definitions. Selecting the Candidate Genes tab reveals a total of 28 candidates irrespective of mode of inheritance. The default is to display all by genes, but you can choose to display by variant as well. The next step is to filter the results by likely mode of inheritance. You can choose dominant, autosomal recessive and compound heterozygous, X-linked recessive, de novo, compound heterozygous exclusively, as well as missing, which displays variants in the candidate genes that are too poorly covered to meet the analysis parameters. Selecting autosomal recessive, homozygous, and compound heterozygous reveals the variant in KCNT1, a known epilepsy gene. Clicking on the variant link opens the genome browser. Inspecting the BAM files for the father, the index case, and the mother reveals an inheritance error that is consistent with the uniparental disomy. Case 13 is a trio consisting of 2 unaffected parents and an affected female proband diagnosed with Ohtahara syndrome or early infantile epileptic encephalopathy. Again, the term seizures was entered into the Phenotype Signs and Symptoms field, calling up 388 genes into the candidate gene list. Selecting the Candidate Genes tab reveals 11 potential causative variants. Filtering on de novo candidates as a matter of course reveals the variant in SCN2A, which is a gene known to cause early infantile epileptic encephalopathy, and was determined to be the causative variant in this case. The CSA reports that for the potential de novo variant, the father is determined to have 0 reads out of 31, and the mother 0 reads out of 23. To determine whether this assessment is correct, the variant is clicked. Inspecting the BAM files for the father, the index case, and the mother confirms the assessment. Case 14 is a trio consisting of unaffected parents and a female index case diagnosed with craniosynostosis, or premature fusion of the cranial sutures. Craniosynostosis and related terms were entered into the Phenotype Signs and Symptoms dialog, and specific genes were added individually in the Add Candidate Genes dialog. To view the entire compiled list, select Candidate Gene Overview. Candidate gene analysis reveals 1 candidate that is rejected as a causative variant. The next step is to look outside of the candidate genes list. The All Variants tab displays all variants meeting the analysis criteria regardless of phenotype association. This analysis reveals 435 variants. When this list is filtered for de novo candidates as a matter of protocol, one variant is brought to light. A missense mutation in the HUWE1 gene, and clicking on the variant link opens the study in the genome browser, and the dominant de novo causative variant is confirmed. Case 15 is a trio consisting of an unaffected mother and father and an affected male proband diagnosed with craniosynostosis or premature fusion of the cranial sutures. The term craniosynostosis, as well as specific genes, were used to populate the candidate genes list. The only variant that emerges as a possible causative variant is a novel de novo missense mutation in the bone metabolism gene DMP1, which is determined to be the causative variant. Clicking on the link and inspecting the BAM files for the father, the index case, and the mother confirms the assessment. Case 16 is a nuclear family consisting of 2 unaffected parents and a male index case and his affected male sibling. The brothers have been diagnosed with mental retardation. Based on the detailed phenotype description, the terms delayed speech and language development, intellectual disability, and global developmental delay were keyed into the phenotype signs and symptoms dialog box. Candidate gene analysis reveals 21 potential causative variants. But when filters based on inheritance pattern are applied to the candidate gene variants, no variants fit the case. Next, inheritance pattern filters are applied, and X-linked recessive analysis returns 4 possible candidates, including the missense mutation in GRIA3, a glutamate receptor known to be associated with autism and intellectual disability. Inspection of the BAM files for the father, index case, mother, and sibling reveal high-quality reads and confirm the stated findings. This concludes this presentation of the Genomics England Rare Disease Cases in the Clinical Sequence Analyzer by Nextcode Health.